soon.
I think we're a bit early, but we'll just wait for everyone to join us.
Yeah, it's only 3 minutes left, so they will join quickly, I guess.
Hi, everybody.
Hi, Matt. How's it going?
Pretty good. How are you?
Hello.
I'm
Still bound to home, or are the children allowed to go to school soon?
March the 8th. So yeah, pretty soon, I think. So yeah, we're all in the house, and we have a workman in the house, and we have a new puppy. It's chaos.
Yeah, for us, the children luckily were allowed to go to school 2 weeks ago.
Oh wow. Okay.
I'm not
Right.
sure who was happier, me or the children.
Yeah, right. I was expecting them to allow primary school children back first and then secondary, but apparently on March the 8th, it's just everyone's back.
No,
which is
here
gonna be
only
quite...
the primary school started up. The secondary school will also be somewhere in March.
Okay. Yeah, we have a lockdown plan that is aiming for normality by, I think, July or the end of June or something, where everything is then fine. Yeah, if all the data are fine, everything's fine. So trying to sort of psychologically help people get through it.
Yeah, for us, they decide by 2 or 3 weeks at a time. But
Right.
we're lagging a little bit behind with vaccinations. I think only 1 million of people have been vaccinated, so that's still 16 or 17 million to go.
Oh, right, really? Oh, that's interesting. I think we're up to like 20, I think we're up to between 25 and 30% have had one jab here. I'm not quite sure how many have had both, but it's going, I mean, it's very fast. It's really amazing. It's like a wartime effort, you know, of huge mobilization of resources. It's really interesting. Yeah.
Yeah, here they're lacking the vaccines. So we don't have all the vaccines yet.
Okay. Okay. I think that also there was some data coming through, which is really interesting about whether or not the vaccines help to slow down transmission, which is, you know, going to be obviously quite important. And the preliminary data is saying that it is slowing down transmission, which would be amazing if so.
Yeah, but here I think mainly the elderly have been vaccinated, so I don't think that will help too much with the day-to-day transmission.
Yes, right. They're not the ones in the pubs, are they?
No, but they are closed anyway, so.
Right. All right. Advance apologies. I need to head off the call at 1:30, just because there's too many other things going on at the moment. So just advance apologies about that. And I'm glad we're all on the same call and we haven't got the wrong times. It's funny because I put in the new ICS files and it seems to have worked okay, but I do have 2 bookings for next month, and I suspect that that's because one of those is legacy. So I'm sure I can figure it out.
I had 2 for this month, but they are both on the same time, so that didn't matter.
All right, fine. But we can always clarify when we get closer to the next meeting.
Yeah, I think when we set those agenda things, we should put the right time A or time B so everyone's aware. So then we don't have to send messages to each other frantically the day before.
Yeah. All right. Does anybody want to kick off with important stuff, or should we just take it in order? I don't mind. Hung, do you want to chair and guide us, or who wants to lead off?
I can go first. We have a little bit of exciting news. So the series 2 stuff, I posted an update on the synthesis for the route that we're sort of working on, where I made a comment saying that the chemical we're still waiting on. So I've done one half of the route to get one of the intermediates. I was waiting for the other one. It actually came in today, so I get to do that synthesis. Wow. And if it works quite well, the synthesis is okay for us to run with first-year students. So we're actually going to do some of this work with, it's called Special Studies Program at Sydney, with some of the students this semester. So I think that it's going to be like around May-ish. And we should be able to do about 10 different compounds. There's a list of analogs we've sort of already looked at, but we're just going to we might make some that are a little bit different just based on some of the toxicity that's related to some of the brominated intermediates. So we may just choose some that are still similar, but just a bit different because they're like category 1. So they're quite like risky. But yeah, so that's one of those things. Thank you. And
so that's that, right? So and it's this. I guess it's some of this chemistry, not the Grignard, right? It must be some part of this.
Yeah.
You're thinking about doing.
So we're just looking at doing the hydrolysis. So the step with the sodium hydroxide and water and methanol, and then the second, So they'll do that one, and then we'll provide them with the brominated intermediate, and they'll do that reaction in the ethanol at reflux to make the final compounds. So it's just those 2 steps. Yeah.
Right, and that allows you to. So you're going to be able to vary both halves?
Yes, so we can vary the pyridine rings on either side of that, and it will provide us with a whole bunch of different ring structures there. So that's what we're looking at doing. And we were just going to look at the varying the pyridine ring on the brominated species. So they'll all do the same first step because that's just easier with all the students in the lab. And then we'll provide them with a different brominated intermediate. So they'll all make a different analog at the end, or the groups will make different analogs.
Right.
But yeah, their first reaction will be the same, makes it easier for them, for the demonstrators, and then the second one will be a bit different. So
that's awesome. Really, that's really exciting.
Yeah.
For this, I guess, so for this compound, this is the one that we had activity on, right?
Yeah.
And I mean, obviously, resynthesizing this is something you're already doing to verify it's active, right?
Yeah.
And I remember when we talked about this, the first time we talked about this, Ben Perry was talking about the idea that this is quite a well-explored motif, which has positives and negatives in the sense that the chemistry should behave itself, and we have access to a bunch of compounds maybe that are already known that we could investigate. I guess in this case, are you going to try and specifically aim for compounds that maybe haven't been made before, or I guess just in terms of the R groups, do we? Is there an easy way of getting into novel chemical space, or do you want to stay in sort of known chemical space for now?
Yeah, so I think we were going to test this synthesis this time around. We don't have a lot of time to do some of the research beforehand, so we're just going to test it with some easily. There's plenty of commercially available compounds that we can just brominate to give to them. But I do think that it's important that we look at ones that aren't commercially available, because you can buy a lot of these aminothiazoles as well. So looking at ones that aren't commercially available and the ones that are novel, and then when we run this next time around, we've got a bit more time to do some planning of what compounds and analogs we actually want to make, then we can look into that. But I just think for now, we need to start ordering the chemicals and stuff like that.
Cool. Yeah.
Alice, no, I haven't. That's the next thing I was going to talk about. Do we want anything more on this? Yeah.
Well, it seems a very nice school project as well. Nice to incorporate it into the teaching as well.
Yeah. So, and the compounds, I think they should both precipitate as well. So it makes it much easier for the students. Yeah. So fingers crossed.
I mean, it's a good point there that if, I mean, the chemistry looks quite amenable, and if things are precipitating, that's pretty nice. It could be, one to engage, schools that have expressed an interest. So, so the Seven Oaks collaboration that we have here has been going on slowly in the background. It's a problem because schools have been closed, obviously. But that's a phenomenal type synthesis, and it brings its own challenges in the sense we need chromatographic purification.
Yeah.
So if this is looking like it's working, that's something that could easily be scaled up, to other schools.
Yeah, and I think awesome. The final compound will, purify that on a column, but the other stuff, it should be okay and they can still make it and characterize it. We're just purifying it at the end to clean it up, right? So, yeah, I think it should be good.
It's interesting, just to mention, I was having another conversation the other day with a couple of people who were talking about how if you've got a pathway like this, of one or two steps going to some group that you're interested in, some structure you're interested in, whether it be, how easy is it to enumerate all the possible variants of the product that you could get with commercially available starting materials, so that you know what's possible already without figuring it out yourself. And it's not easy to do it because the number of possibilities gets very large if you have two-step synthesis like this, and if there are multiple starting points that you can use from catalogs, you start dealing with millions of compounds that you could possibly get. But it's an interesting one, because this might be a nice case study to go for about how it's possible to do complete enumeration of a virtual library of those products with every possible R group. To spot things that might be of interest, so it becomes like you know what I mean by the enamine real space library is a virtual library of everything that can be made with commercially available materials with an amide coupling, and it's billions of molecules, right? And you can sort of browse, I'll have that one. In this case, this might be a nice one to go for as a sort of bespoke new virtual library of things that could be made. So yeah, it's nice from that point of view as well. It's quite straightforward,
Yeah.
Great.
So then there's another thing that links into this, Alice's two things she spoke about. So we're also hoping to run a workshop over our Easter period that might be intensive. That's for third years who were in second year last year, who because of COVID haven't had any chance to do organic laboratories. So they could come in through the break, and we could possibly run this synthesis with them as well. And so we might get some more of that. we have to get permission for that first, and that's related to COVID. So we'll see how that goes, but just so you guys know. And the other thing was that we had a chat today with, I can't remember her name. Sorry, Alice, you'll have to help me. I think Christy or Caitlin. She's from Auckland University. Caitlin, there we go. She's from Auckland University. And last year, because of COVID, one of her co-workers actually got really into Data Warrior. And they did a collaboration with another project that had some like in vitro data for a bunch of analogs. And basically the students just played around in Data Warrior and did some structure activity relationships based on the in vivo data that they had and some other interesting things. To which that project has now stopped, and she's looking for something else she can do that's very similar. And we were saying that this might tie in really well with open source mycetoma, and we've been talking about doing these types of things and looking at different trends between different properties of the molecules, and also looking at structure activity relationships. So we had a chat with her. She seems pretty keen on that idea, and we have plenty of molecules to look at. Like they could look at some from the pandemic box and things like that as well, that they might be able to find some trends that we can carry through. And hopefully, if they do some of it for the aminothiazoles, the series 2 we were talking about, they could suggest structures for us to look at making based on the modeling of what they think might be more active. And then we can carry that back through with the students at Sydney that then do the synthesis that could synthesize what they've suggested, and then carry that through again and again. So yeah, we just had a brief chat with her today about that. To me, she seems pretty keen, so we just wanted to share that. She's going to join on GitHub. Because she's in New Zealand, it's like 1 a.m. for her right now. So it's a little bit like too early for her to join for us, but maybe the next meeting, she might be able to come along to that one. And we can chat a little bit more about that, but yeah. So that's something else that we're also looking at doing. And we've talked about doing some of this stuff before, so we can pick a starting point of what we think is most important for us, and we can go through that with her and stuff like that.
Kimberly, funny enough, I was approached by a teacher I know from a college in Breda, which is in the south of the Netherlands, for some students who are looking for a sort of similar project as well. I will speak to them this afternoon as well. So there will also be some students from that school probably looking into the databases. So it would be nice to see what the students from Auckland come up with and what these students come up with, to see if they find similar features or if they are focusing towards different pathways.
Yeah, that will be cool. We also talked about that if you had groups working on the same set of compounds and seeing like what relationships they found and seeing whether it was the same or different. And yeah, so that sounds really cool. We can have a big, nice connection, international collaboration.
Great, it's all happening.
Okay, so I'm just gonna continue on with the items from our agenda. So I've already uploaded the previous recording onto YouTube, so that's all good. That's been fixed. I've also done a little bit of chemical structure. I'm just gonna get. So this is for just to try to do a bit of structure and activity relate analysis. Just let me share my screen. Can you see my screen? Just make sure I've got it correct.
There's 3 windows up.
Oh yes, that's
yeah.
So I've just did a bit of ChemDraw, and basically what I did is I had to look through my set of compounds first, and these one in the boxes are the one that didn't have high activities in terms of in vitro, so they don't have high IC50 and 90 and MIC values, which didn't carry them forward to the in vivo. And all the rest of these are the in vivo activities from the previous update that the previous in vivo update that Wilson had. So basically, we just these ones are what it looks like is for this motif here. We do have so in this part here on the ring, we actually have, if it's too big or if it's too if it gets chopped up, and actually will lose the activity. So these three are the three that went on to be in vivo tested, which was in last meeting, and all of these other ones, I just got these from the our biological evaluation on GitHub. So it was this one down here. So I'll just put it in here. So I just did a skim through and I saw it. So if you chop that up, it actually just lose the activity as well. So we kind of like want something in this range here for this motif, and you actually have other things sticking out in the ring. It will also kind of lower the activity as well. So that's what I found for this particular structure. And for 12, 14, so I was going to ask, I didn't have a chance to ask Wilson as well, just to probably rearrange it. So we've got the 30, 14, and 12 in the one graph, and then the other one in the other graph. But yes, that's what it is. Another thing is for the middle, so the alcohol, so you just remove the alcohol reduce it down to just a hydride, and that will actually lower the activity as well. And I think this probably has to do with the electronics, so the polarity of the motif, whereas you can see if you have the O-methyl and the amine, it actually was good enough to proceed to in vivo as well. And there's one more down here that I found where this was from the master list. So 37 never made it to in vivo because the in vitro was low, and that's just the difference between these two. So that's some of the things that I found when I just basically went through to have a quick look at what we have so far.
So that's all your compounds in vivo?
Yes, so in vivo and in vitro, except that one. That one's not mine, but yeah.
But I mean, what I mean is that all of the compounds that have been evaluated in vivo are on that picture.
I do not have Dimitri. I haven't.
No, but I mean, they're all yours, right?
Yes, yes.
And so the ones that are inactive are the ones that are in the boxes.
Yes.
Right. Okay. And in terms of quantifying, so this is a question for Wendy and Wilson. In terms of quantifying this, is it possible to quantify the activity using the area under the curve, or is that silly, or would that not tell us anything?
Well, we looked at the area under the curve and the percentage survival. Of course, they are closely related to each other. It is possible to make a figure like that, like we did in the previous one, where we plotted percentage survival against any feature we wanted to know. I'm not 100% sure if it will mean much, because as you can see, the lines are very closely to each other, but we can always try. Theoretically, yes, it should mean something, because the higher the percentage survival in the end, usually the higher the AUC compared to the controls. And I agree, we need some kind of quantitative measurement. Unfortunately, we were not able to do all the histology, etc. And we decided beforehand that that might also not be necessary for these experiments so far.
I just wondered what your... Sorry,
yeah, this is what we have. So let's do it like that and see what comes out of it. But if it doesn't demonstrate too much, we can always put that into the discussion.
Yeah. Yeah, I just wondered what your preference was for the more meaningful number. You know, so all the larvae eventually die. I remember we talked about that last time, that it's early on in the treatment that the impact is going to be had. And so I don't know if you wanted to choose a percentage survival at a certain day, or a percentage survival at the end, or just the area under the curve, and whether you had a preference for what you think is telling us more.
Well, the area under the curve, of course, takes the whole curve into consideration. So if you have a good effect only in the beginning, but that diminishes later on, you will still have some effect in the AUC. So I would be in favor for plotting both, actually the percentage survival and the AUC, and compare that, because with the AUC, you will have the whole line into consideration. With the percentage survival in the end, you have the final outcome. So those are two values, and then you can take both values into consideration. If we want to plot another day, that's not that difficult for us to do anyways. Then I would say that would be day 4, the first day after treatment. So larvae are treated from day 0 till day 72 hours after. So then the most meaningful other time points would be day 4.
Do you, from these curves, is it simple for you to calculate the AUC?
Yes, it's actually one of the features of the program. So it's just hitting one button.
That counts as easy, yeah. Okay. I mean, yeah, this is the crucial thing. So if we have, what we need are these molecules that you're showing there, Hung, with Dimitri's and any other that we have tested, of course, in this series, in vivo, with the structures, obviously, with those AUC values, plus any physicochemical parameter that we need to investigate, like log D. That's what we're looking for. So Dimitri is looking into this, the log D values also, so that we can plot those versus each other. And if we're lacking any, we may have to ask Ben very nicely if he wouldn't mind calculating some of those for us, because it would be useful to have them calculated the same way throughout.
Hi, Matt. It's Ben.
Hey, Ben.
Hey, I can do that. It takes me less than 5 seconds to generate those. Right. What I need is just a list of the smiles. Yeah,
that is awesome. Yeah, okay. I mean, we just want to have one set of values, and then if we see this trend that we were suggesting last time as backed up, that in vivo is better served by a certain log D value, then that's great, and we can keep on using that as a design criterion. If not, we look for something else, or we can look for something else anyway, but we just got to answer that question. So yeah, this is obviously important for that, but I think that the AUC value would allow us to quantify very nicely what we want to see.
Yeah, so what if we generate the AUC, the percentage survival at day 10 and the percentage survival at day 3 and day 4? So day 3 is still in treatment, day 4 is the first day after treatment. Then we can always cross-ref that with the values we want to generate, and see which one is the most predictive to use in the final version of the paper. Right. Or maybe it will be 2 values, or maybe it will be all 3 values, because they will demonstrate different things. But I think those are the most logical time points. And then because we're not chemists, and if they are plotted like this, I can see that there are differences and similarities to molecules. But if Hung, for instance, wants to have some molecules grouped, please send us the list which you want to have grouped in the figure, so we can change the figures in that respect. Because for us, sometimes it's very difficult to see which is chemically similar and which is less similar, because it's not our expertise.
Yeah, definitely. I can do a list over after.
So I'd encourage the values that we have for AUC and survival at certain days. It'd be very useful ultimately to have everything in the master list, right? So that we don't get confused. But for plotting the graphs, we will need to extract those so that we just plot the values that we want. So Hung and Dimitri, I guess you can work together to try to assemble, to make sure that all of these compounds, of course, are in the master list. You could then extract those into a sheet for Wendy. So you've got the myos codes, and you've got your internal codes and the smile strings. And then you could maybe forward that to Wendy, and they can insert the values for the survival and so on. And then we can use that same sheet and send that to Ben so that he just has a list of the smile strings, and we can get the predicted log D values, and then we're done. Yeah, is that all right? So
about the molecules which proceeded to in vivo stage.
Yes, this is all about the in vivo test. Yes.
Because I wanted to assemble a graph today of all the molecules that were tested in vivo, but because of the lack of the log D values, so I couldn't generate the graph. But there are around like 20, more than 20 molecules, around like maybe 22. I remember the exact number.
This is Ben. This is where keeping data in tables rather than in pictures is much more useful, because if you have all this in tables, you can send me, you could send me the file now, and I would get the log Ds back to you in under a minute, because it's a click of a button. But if it's everything's in pictures like this, you have to go through them one by one, and it takes ages.
Yeah. So a sheet
I think I saw that as an action from a couple of meetings ago, maybe, that we need to keep as an absolute priority. I would advise keeping the data set, the data sheet or Excel table as the priority thing to keep up to date, rather than pictures like this.
Yeah.
That's a picture spreadsheet like that, I can understand why that's a nice way of looking at things, and it looks good, but it's not very useful for managing projects. So yeah, so as long as this kind of sheet, as long as that is the thing that's kept up to date first, and everything else stems from it, then it makes it easier.
Yeah,
but you see that column H with the smile string on it. If you just send me that, and I can put in a new column that says log D next to it straight away.
Great. I mean, I'm thinking about the way in which it's going to be easiest for Wendy's team to put in the AUC value. So an option is just to highlight the compounds here that need those, which would be fine, or we extract them out. I don't really mind which happens.
I mean,
we can add this. We can add it in this table. That will not be that difficult.
Great. Okay, then let's keep it like this.
For the Excel file, are we adding an additional column with log D values?
Mm-hmm.
Like say after the IDs number, or
somewhere logical.
Just for it to be like in the same place, all information set.
Yeah.
Carry on, Ben.
I was going to say, if you send me this file, the file is the up-to-date one. Just make sure it's the up-to-date one, and I can send it back to you with a new column in it.
That's great. Yeah, perfect. Without
don't put the column in. Don't put the column in yet, because it will be. I mean, you can do, but.
Okay,
it's
that's
not
perfect.
done by hand. It's just it'll. I just need to open it up in something like what we do it in StarDrop probably.
Okay, wonderful. So for plotting the curves, we're extracting a set of these values, but for the sheet itself, as Ben said, keeping that as the master source of everything is really good without fragmenting it. Is great. Yeah, and if you guys, Wendy and your team are happy to put them directly, that would help a lot. All the code
Wilson is chatting in the chat function, and he says that the data is already there. Look in the in vitro and vivo potency and log D values. All compounds that work in vitro and in vivo are placed there. There's a column for log D values as well.
Okay, so it would be very helpful to try and merge those if possible, just because if you fragment the sheet, it becomes more difficult to keep track of things.
So there's actually two chapters as well for them as well. So there's this one and there's that one.
One's in vitro and in vivo. The other one's only in vivo.
Yeah, but I mean, it's a bit fragmented, as Matt just said.
If it's possible to merge, that would be preferable. I know it's a pain, but it just, people then start adding new data to only one tab, and we don't then have a complete data set for everything. So that would be good if we could try to merge if possible.
So do you mean merge it to the main one, so the master list? Is that correct?
Yeah.
So to this one.
So the MyOS compounds, if the in vivo, all this in vivo data and log D values can go in here.
Yeah, I think that will be a good idea as well.
Can you go a little bit below in your screen so that I can see the tabs?
Yes, just give me one moment.
You can't see the, I can see the tabs there, Wendy. You can't see that on your screen?
Yeah, now I can see. So are these, yeah, I will, that's why I will ask Wilson to do that. I think they are on the, usually all the data should be, of the Fennery moles should be on the EpiChem library sheet, or are all data, because the pandemic and the pathogen box and the stasis box are here as well? Or do you want us to merge everything in one sheet?
So there's a tab there for the EpiChem library, and we can keep that separate because that's pretty large and predates what we did. But for everything that we have generated since the start of this project, it helps if it's in one sheet, really, just because then we're not confused about where things should go. So ideally, yeah, everything which would go in the sheet. We can start with the stuff that we're talking about now, which is the in vivo potency data for the Fennery series, and just make sure that that's in there. And then for the pathogen box and stasis box, it really helps, yeah, if everything can be in there eventually.
The ideal is to have a database for this rather than a spreadsheet.
There is an ideal database.
The problems everyone's arguing over. But,
No, I meant because now we have the pathogen box and the stasis box, and I know that the data for the in vivo should be in those tabs as well, because I generated those tabs in the beginning of the project. But I can imagine later on, because now we have one for the pathogen box, then we have one for the stasis box, then we have the pandemic box. Ma is going to generate the COVID response box. That it will also be easy to put everything of those also only in one tab and not in all tabs, because if you want to look for something, you don't need to browse all the different sheets.
Maybe we can add another column that has an origin. So it could be the pathogen box, so we know where it's come from, and then we don't have to have a sheet for each. And if people only want to look at pathogen, they can just filter the table based on the origin.
Yeah, there's already a column for by whom, so that can be your filter if you want to. Pathogen, pandemic, stasis, that kind of thing.
Yeah, I guess we can just put stasis box or the EpiChem library into the by whom section as well. Maybe just
whatever. Yeah, yeah. Like I said, it ultimately it helps, just because then you can do one search in one place and you never worry about where things should go. That's all. Yeah,
because for me it was wondering, might it not be easier to put the EpiChem library together with the Fennery mole analogs? Because if you want to browse any structure which was made or screened with the Fennery mole backbone, it should be still in two. It's currently still in two different spreadsheets.
Yep. So so it would be great if people want to take this on, because I think that would help a lot. As soon as you do that, just be aware that you will need to generate the MyOS codes for them, which is not, it's not completely trivial, because you need to worry about salt forms. That's all. So the code assignment is easy, but the salt form, you just got to keep an eye on it. So if there are hydrochloride salts, right, you've got to have the right salt code for them and things like that. But it's not impossible, just a bit fiddly.
Yeah, but for now, if for instance, if we are going to merge this in our lab, we cannot, of course, generate smiles. But if we simply merge and leave those places empty if they are not there on the EpiChem library, but I think they are, then that makes it also clear which data we are still lacking if we want to compare later on everything with each other. Because I can imagine that in the final manuscript of the Fennery moles, we want to include our previous data as well for the final analysis, just to get a little bit more power into the equation.
Yeah, yeah, and you know, just be aware that the full, the full epichem library hasn't been screened against mycetoma. It's been screened against other things. So of course that part of the spreadsheet will be blank, but so it's a little confusing, but of course we all know that the compounds are kind of available and have a history here, and they are phenomenal analogs. It's just they don't have any in vitro potencies that we're interested in. So the spreadsheet just has to be able to take care of that. So I mean, I maybe put that last on the to-do list here, where the first one is the in vitro and in vivo potency that you've generated, and make sure that's in there. And then we can think about merging the boxes because they've been screened by you, and then maybe the epichem library last in terms of a sequence of events.
Okay.
Yeah. Okay, sounds good. Just so sorry, I have to run to something else. I've got to, unfortunately. I just wanted to mention one other thing here, which was thinking about the results from some of the box screens that you've done, Wendy and team, and about what we should be doing about those results. Because, you know, I mean, the phenormals, they have a known mechanism of action, and some of the other compounds that we're talking about have known mechanisms of action. And it just feels to me as though very generally the big wow factor of some of the results you're generating is for us to highlight compounds which have potentially novel mechanisms of action, unexplored mechanisms of action. And I suppose what I was looking back at some of the screens that the data that Wilson posted, I think, on the pathogen box stuff, which identified, you know, olorifim and things like that as potentials. And some of the benzimidazole compounds, I think they were. And I just wondered if we could, it would be very useful to have an analysis of, I'll bring up the page that I was thinking about, if I can just share screen, just so that everyone's on the same page. Yeah, the pandemic box analysis, right? So this, oh no, I think I've got the wrong thing. Wait a minute. I think I'm in PowerPoint. Yeah, sorry, hang on one sec. Yeah, so the analysis that Wilson posted of some of the compounds that you found here, you know, these are color coded according to continue to MIC and no further testing and so on. And I guess I was interested in some of these structures. And really what I want to know here is whether or not some of these compounds with which I'm less familiar, so for example, this MMB compound here, whether they have known mechanisms of action, and whether these are mechanisms that haven't really been explored particularly well as antifungals, or, you know, obviously particularly well as anti-mycetoma agents, but something that's a bit different and a bit new. And it feels as though maybe we should be keeping a very close eye on anything that comes up from your screens, Wendy, for compounds that maybe have a potentially new mechanism of action. So it would just be very helpful if people already knew the mechanisms of these compounds, if that could be highlighted in the analysis of this box, so that we could sort of focus in. So there's a class thing here, for example, but, you know, for some of the molecules we don't have anything. And it seems like when something does come up later, we should actually zoom in on that and say, actually, wait a minute, that maybe has the greatest future, generally speaking. So for this example, for the blanks here, I just wonder if we could try and fill those in, if people wanted to try and do some literature searching to try and fill in the blanks a little bit, and we could think about highlighting some of those more. Someone was, sorry, Wilson, were you talking?
Yeah, can everyone hear me?
No, sorry. And, you know, Wendy, I know that you've had an interest in the DHODH thing as well. So I don't want to tread on your toes if you're pursuing that separately, but, you know, looking at that page, that I immediately see that and I think, hang on a minute, we haven't explored that at all. Maybe that's where we should be looking for greatest impact, I suppose.
Yeah, for the DHODH inhibitor, yeah, I know that Meg Phillips' lab is looking into that as well. Yeah, so, but apparently she's trying to express the enzyme and it doesn't work very well. So the enzyme of Madurella mycetomatis is a little bit different. But I agree with you that we do have still some unknowns here. And some of the MMV compounds we cannot find any literature about currently. But I guess that Ben, who was on the selection committee of these compounds, or Jeffrey could fill us in on that. Unfortunately, in this box, we found a lot of azoles, and we do know the mode of action. And we also know that raviconazole is currently, well, fosraviconazole is currently in the DNDi trial. So I agree with you that focusing too much on the azoles is not giving us that much more insight. It's just one azole or another azole. So I also would like to go into the new classes. And then I think we should look at those which showed some activity in vivo and see which have a new mode of action. And olorofim, that should be a clear candidate also maybe for further investigations in vivo. So what we aim to do there is to look at more concentrations and maybe combination therapy, at least in the larvae to start with, because that's already relatively well advanced, and that molecule might go into clinical investigation later on or something like that, because that molecule is already quite far advanced in development, of course. But on the other hand, I would be more interested in modes of action which are not well studied in other fungal infections as well, because that will really give us a new edge, not only for mycetoma, but also for all the other fungal diseases and fungal skin entities especially. So what also interested me was that from the benzimadazoles, we had one very active one, and the other one was not that active in vivo. So that might also be something to look into, and it should be a different mode of action than the azoles, because when they look at other fungi, it's not an ergosterol inhibitor. It had a different mode of action. And then we should ask, of course, people from MMV if they have some literature about the other MMV compounds where we found prolonged survival, but not much literature, because if that will help us to find some idea of the mode of action, maybe we can also look into it, depends on what the suspected mode of action is, with different approaches, maybe even to identify the mode of action ourselves. We're doing a small trial currently with collaborators in Japan, where we take known antifungal agents to see if we can use a transcriptomic approach to get some clues about the mode of action. We do that, for instance, with the azoles, but also with olorofim, because we know what the mode of action should be. And if we can get that out via such a system like that, that would be awesome, because that would also give us means to identify unknown modes of actions currently. Normally, you would do this by generating a knockout library, but unfortunately for Madurella mycetomatis, there's no knockout strategy currently. But we're still trying to do that, and we are able to introduce, for instance, GFP into Madurella now, but we're not able to knock out specific genes, because in fungi, that is sometimes a little bit difficult because it integrates randomly into the DNA instead of targeted to the gene you want. That's why we cannot do the classical way. But if we can do it via transcriptomics, for instance, that would give us an opportunity to identify new modes of actions based on the hits we have, even if there's no literature available. And since we are testing that this year as well, I think that will be a new avenue for this project as well, because that would allow us to explore even molecules where there's nothing known in any other organism as well.
Yeah, that's great. And so, I mean, for the ones on this slide, you know, which have the blanks, if MMV and/or collaborators don't know what the mechanisms are from other studies, then maybe we should prioritize these ones in the assay, the transcriptomic assay you just mentioned, because to identify something new here would be amazing. You were already in touch, Wendy, right, with, I think, to get about olorofim analogs or DHODH inhibitors already. If that was through MMV people, I wonder if you would be able to reach out to them about these blanks, whether they know anything about them, or did I get the wrong end of that?
I will make a list out of those based on our in vivo results, and I will email it this afternoon, because yeah, I just spoke to Jeremy yesterday, so I don't think he will mind to send me that if he has the information. Okay,
fantastic, great. Okay, and I just wanted to flag it up before, given that I know you're screening the COVID box, is that right?
Yes, that's Ma's project.
Okay, fantastic. Because I mean, the same thing, right? So if we get a molecule that has some effectiveness and which has a mechanism of action which we're surprised by, we just all our attention should be on that.
Yeah, unfortunately, of course, with in vitro we know earlier than with in vivo, but yeah, here we're in February, end of February, so in a few months' time we could try to screen the hits we have. So. Right. But that's also one thing we still needed to do with the log-D. But if I saw that all the smileys were there, because then we could cross-compare with the in vitro activity as well to see if there's a different subset of values needed for in vitro penetration.
Mm-hmm. All right, great. I'm sorry I have to run, but I'll catch up with what happens afterwards. Don't give me all the action items, okay? I'll see you next time. All right.
Yeah, man. I'm just gonna have a look at the rest of the action items that that were on GitHub. So the next one is actually, is there any other updates from Wendy and Wilson's from the previous time?
Is my mic working now?
Oh yes, it is.
Yes. I'll finally I'll share something I wanted to tell you guys. Can everyone see my screen now?
Yeah.
Hello?
Yes.
Yeah. Okay. So this was what Matt was trying to find out. So we have tested 14 compounds from Ghana, but unfortunately none of the 14 showed any in vitro activity, as you can see, highlighted by the arrows at 2 concentration, 100 micromolar and 25 micromolars. The growth rates were 90 to 100%, or that there's 115, but it's something that is not really good. We are looking for growth less than about 20%. So yes, none from Ghana actually worked. And in line with new molecules and new structures, what Matt said. So if we look at this 8-aminoquinolone compound, they have 3 derivatives: primaquine, tafenoquine, and palmaquine. So primaquine was previously tested in pathogen box. The IC50, meaning the growth was at 25 micromolar. As you can see here, it's only about 64%. It's not what we want. We want something about 20%. This is primaquine. However, as we all know, tafenoquine here, it worked sort of okay in vivo, although there was no significant difference. it was still, and tafenoquine was also able to inhibit all the other mycetoma causative agents. So if you look at down here, because they're the same derivatives of 8-aminoquinolone, tafenoquine has a bit of a more complex structure than primaquine or palmaquine. So I was thinking, because I know not much about chemistry, I was thinking maybe is there a clue we can get from the tafenoquine structure to why it was able to inhibit growth more than primaquine, for example. Maybe one of you can sort of say something. I have no idea. So that was one of the questions I have, and maybe from this we can also see, okay, maybe this is what makes the compound able to penetrate the grain and
perhaps it's enhanced lipophilicity because of the trifluorophenyl methoxy group.
Yeah, the 3-fluorine. So that's just been previously shown in the phenaramo analog, that the CF3, the bromine, the chlorine, so like all of the more the halide or the halogen add-on actually does.
So this bit, yeah?
Yeah, that bit.
Yeah.
And I think, Hank, you had this in your previous work as well, like the trifluoro group.
Yes.
In some of the compounds, I think, if I reckon correctly.
Yeah, and previously I just showed on my screen before when one of the compound got, we just cut off one of the halide, it actually just dropped the activity down as well. So I'm guessing the most likely is that CF3, so the 3-fluorine on that end there.
Okay.
On the ring.
Okay, yeah. All right. It's really interesting to know. We have to look into our previous data to see if those compounds that actually worked in vivo, how many of them have these three-ring structures?
Also, we could try and maybe see if we can get an analog of that with the SF5 group on the replacing the CF3. That would be a lot more lipophilic. It would be interesting to try.
Yeah, could be. So yeah, let's see if we can find more of these in the compounds that we have tried, and then maybe there's a link to that and we can put it in a manuscript. Like I said, it's your forte.
Yes, I'll get a list of what I know personally that has that probably has either good in vivo or in vitro. I'll put together a list for you with what I know.
Thank you, Hong.
This is the type of stuff This is the type of stuff we can do with like the students that want to do some of this like structure activity relationship stuff. So like if you have any questions like this that like pop up, like keep sharing them with us, because they could sit down and look through our libraries to find compounds that have that group and look at the relationships. So like if you have any other stuff like that, let us know and we can share that with them so they've got a bit of direction.
I think that would be great. Yeah, especially with the like the Epichem library. There's like 700 of them in there, so have more eyes to look at it would be a great idea.
Yeah.
Just to jump in here, right? So just because a compound has a CF3 group in it doesn't mean it's going to have activity, right? You have to take the structure as an absolute whole. It's not the presence of little parts of the molecule that all add up to have different activities. So, I mean, yes, you could look through the Epichem library or others to see compounds that have CF3 groups, and you will find them, but that doesn't. There won't be any relationship between their activity and this compound because of the CF3.
Yeah, I was thinking more like the core structure and the functional groups that are coming off that core structure, not
Perhaps
specifically just the CF3.
Matters more into phenolquin than it does into phenaramols.
Okay.
Kimberly, so you would be using Data Warrior for that?
Yeah, so the students at Auckland University were using Data Warrior last year, so I assume that they intend to use that again, which works in nicely because that's also what we use.
What if instead of mining for a functional group, we get all the lipophilicities of these compounds and plot them?
Well, we can do that too. Like we can ask them to look at whatever properties of the compounds we want to look at. It doesn't have to be structure activity. It could be anything.
Yeah, there could be, because there could be an island of lipophilicity that we need to be in for these things to kill.
Yeah.
Or it could also be the difference between these three maybe log-D values. I don't know. it could be that that group just dropped your log D a little bit or something like that. That remains to be seen when we have the data. Right?
It'll raise it a lot. But I would imagine that it's higher log D that you're going to need to be able to get grain penetration, not lower log D. So that Anthony's point is actually bang on. There is a hope. What we're hoping for is that there is an island. It's a good term. I like that term. There's an island of lipophilicity that you need to be in. So the molecule still needs to have the bits, the functional bits that are hitting the target. In this case, I would guess on these three compounds, it's the 8-aminoquinoline. That's a known motif, and then everything else that's on the molecule is just there modulating its lipophilicity. And when you are in a certain area of lipophilicity, that allows the compound to get where it needs to go and have its impact. The problem, and one of the things that we're going to run into almost certainly, is that usually when the lipophilicity is you're going to be needing, I'm guessing, higher lipophilicity to be able to get these compounds where they need to go. And the problem with high lipophilicity is that also usually leads to bad toxicology, it leads to poor pharmacokinetics, it leads to all that sort of manner of negative things. And the question is going to be, can you, is there an overlap? Is there a Venn diagram between those two islands of good drug-like properties and ability to get where it needs to go to have an effect? And if there isn't, then this is a dead end, because you're never going to get a compound that has both. Right? That's the sort of thing that happens over and over and over again. I mean, we see it in parasitology all the time. But,
yeah, maybe it's a little bit like with amphotericin B, where you also have two different sides to the molecule, where the one side binds to the ergosterol and the other one makes the pore. Because the overall value will be different, but if you look at the different parts of the molecules, one part will actually bind to the ergosterol itself, and the other one is not binding the ergosterol, but producing the pore. And that makes it also difficult when you don't know where it's actually binding or what the target in the fungus is.
Yeah. Yeah. So the data that I've put on the sheet, they're all updated so far. In vivo and in vitro, everything's updated, and yeah, I think the next thing we have to do is look for similarity between compounds that work in vitro and vivo. Just a bit of a clear up for what we have discussed earlier. My mic didn't work, so I couldn't contribute. So in the Excel sheet on GitHub, the extra tab where I've renamed it as in vitro and vivo potency for log D values, that sheet is just to make it easier for Ben to sort of calculate and key in the log D values, because the compounds listed on that sheet and are ones that I want the log D values for. So that's just to make it easier. And when the log D value is being calculated, I will then personally put into their respective sheets. For example, pandemic box, epichem to epichem. So yeah, that was just what I want to say. And also thank you, Ben, for sorting the log D values out. Yep, I have nothing else to share.
All right. I'm just going down the agenda again. So I think we've talked about most of the items on there. So we've already talked about Kimley's series 2. We talked about darabin, difenoquine, and the DHODH inhibitors. There's one that says there's any need to go back to epichem to source the remaining pheneramals for the paper. That was one of the items. Any need to go back to epichem to source remaining pheneramals for the paper. So anyone has any idea about that particular item? No. I'm actually not sure what's there.
I think we need some more information on the relationship between the log D with the in vitro and in vivo data, and then go back and see if there's any compounds from the epichem library that might fit what we think will work well, to which we have those compounds so we can just call them in and then test them and see.
Exactly
that's my
what
assumption.
Matt
But
discussed yesterday about log D correlation with the in vivo data. Because we need to decide like what to do next in terms of like synthesis as well, like see the trends. Can I, Hankar, do you want to show something, or I can show the screen? I don't hear you.
So you can go to Matrix. Screen.
Yeah, just to come back to the question that we discussed at the beginning. So what I did is just like I looked at data for the compounds that I've made for the thesis for the upgraded report. But basically, what we need for sure is to have all the in vivo data to see the correlation between log D and percentage survival. So this table refers to the in vitro mainly. But then again, like I wanted, just give me a second, please. Yes, so basically those are all the compounds that showed in vivo potency as well, that Wilson updated. But I have a question. I don't see all the data points in this row. Is there like some sort of gap, Wilson?
So if we don't have data points in the under that column, that means we didn't do any in vivo work for that.
So when I'll be creating like a plot graph, should I just omit those compounds, or how should we? Basically, the idea was like basically just to collect all the in vivo data and then to see the log D correlation with the larval survival as one of the graphs, as one of the counts.
So I think, yeah, for compounds that we didn't do in the larvae model, I think you have to omit the in vivo part, I think, because we don't have any data for that.
So basically, this table that I'm looking at is final. This is the final set of information.
Yes.
And it won't be updated like in the future.
Not the ones that we have tested in vitro for. So the ones that we tested in vitro and did not work in vivo, I don't think we'll test the ones that didn't work in vitro in vivo. You get what I mean?
I think my understanding is that so when this team always tests in vitro first, and if it passes in vitro, it goes in vivo. So if it's like larger than 16, for example, that would mean that it definitely did not pass in vitro. So by principle, it will not go to in vivo. Is that correct?
Yes. Precisely, yeah.
So we do have a cutoff as well for in vitro. So some compounds will have some activity in vitro, but not enough to go into the in vivo system. And that's simply, it's not because we don't want to, but it's because the throughput of in vitro is of course much higher than in vivo. In vivo takes us a lot of time, and therefore only those compounds which we think have the best probability are selected for in the larvae work. And yeah, that's why we don't have for everything. But I think if there's a survival percentage, the area under the curve is sometimes missing here. Mm-hmm. So that needs to be added, I guess. And next to that, we will also look at the survival percentage on day 3 or day 4. We will look into the data, which one might be best suited to do.
Yeah.
Okay. So yes, that answers the question. I wasn't sure whether like some data points are missing or this is the final one, but now it makes sense. Also, I have a question. Do you know for the mean percentage growth survival of the pathogen, so number like 25.4, should it be considered as medium fungal growth or like a low fungal growth? Like what are the cutoff points for?
Cutoff points is normally 20%.
20%.
Yeah, 20%. We say that the compound was potent in.
So everything below 20% is basically green. It's like low.
Yeah. So the ones highlighted in green.
Okay.
See there, yeah.
And also, I noticed that last time when you were referring to the potent compounds with regards to my synthesized compounds, you said like that only 2 are potent, but according to the Excel sheet, there is also 25.1, DM25.1, which is also potent. And
Yeah, I think we DM25. I don't. Yeah, we did not test it in vivo because the MIC value. Oh wait, no, did I?
Because that's the reason why I'm asking, because the MIC value, like for DM7, yeah, which was potent, is the same.
Okay, now I got it. So for this time, we took all compounds that worked under MIC 58. And the reason I took DN 7.1 is because you sent us both twice, and one of it was lower than MIC 50. One of it was lower, MIC 50 was lower than 8. So that's why I ran DM 7.1 again.
Yeah, maybe if you go up to look at the value, because here DM 25 was 8.1. Oh no, that's 25 micromolar. Sorry.
Yeah. So yeah, DM. I took the cutoff point for in vivo work, and that was for MIC 50 below 8, not counting those at 8. And okay. DM 7.1 was put in because you like I said, you gave it twice, and the first one was below 8, and the second one was 8. And the first one I named it P4 something something 003. That was the MIC 50 was 4, I think. That's why we took it for.
Okay. Yeah, because I got confused because they had like the same values, but now.
Yeah.
No. So it should be below 8 at least, like for B.
We for this one we took 8 because we had lots of compounds that were below 8 already.
Yeah, so that MIC 50 value is sometimes a little bit arbitrary. So if we have, for instance, all compounds with very high MIC 50 values, then we have this cutoff of 8. But if they are all very low, then we sometimes use a lower value. It's just for us to narrow down the compounds we need to screen. What is physically doable and what is biologically sound. Because sometimes if we would use too high of a cutoff value, then we can't screen them all. And if we have many with a very low, then we can't screen them either. And if we have an MIC 50 of 8 already, that's quite high if you imagine that we are injecting into the larvae that will dilute into the hemolymph, of course, itself. So I'm not sure if you can even reach 8 microgram per mil in hemolymph if we do a simple calculation, not considering binding to proteins in the hemolymph. So that's maybe something for you to calculate, Wilson. I don't know by head if that was what the actual amount is in micromolar we can reach in the larvae if you don't take into consideration binding to proteins.
Yeah, I can look into that, yeah.
Because that's a little bit where that cutoff value was based on.
Yeah.
Probably that's it for me.
Okay, that's great.
Okay, we discussed everything.
Okay.
Oh, and Dimitri, based on your question, which is medium fungal growth, we didn't set a clear definition for that, but I would say somewhere between 25 and 75, I would consider medium. 75 is maybe already a little bit on the high side. Yeah, I would take it a little bit.
I want to make sure that like we're consistent with this because we already have a first Mycetoma paper, and I think, well, yeah, what's the gradation? That's the reason why I asked this.
Yeah, I think we only did below 20% is inhibited, and the rest is high, but I'm not sure if we really made a medium value. I don't think so. Because if you look at, for instance, DM17.3, you can see that at a high concentration, we even get higher growth as a growth control. So that could indicate that at high concentrations, it might stimulate growth even. But we never looked into that in that depth into those data.
Yeah.
Wait, but Wilson said just like minutes earlier that like everything below 20 is green, and now you're saying that medium is 25 to 75 is medium, which means like if I have a number like 22 or 23, does it mean that it's still like low fungal growth?
Yeah, I don't know why this one was green, but I think in the original database it was below 20. Below 20, wasn't it, Wilson?
Yes. So anything growth below 20, we can say that the compound is potent against the organism.
I see. Okay. Yeah, that's really
also
useful. Thank you.
Yeah, like what Wendy said, we haven't really defined what's the medium fungal growth. We until now we only had below 20 or more than 20. Yeah.
But 75% is like quite a high number. Like, how can it be medium? It's like more than a half. Doesn't that mean that it's like already not working and it's like not potent enough?
Yeah, we based this on our experience with itraconazole, with amphotericin B, etc. And then we saw always that below 20%, it was corresponding to no growth. We took 20% because Madurella mycetomatis produces a pigment which can interfere with the dye we use to test viability. So that's why we use 20% as a cutoff instead of, for instance, 5% or 0%. And that's simply because of the pigment secreted by the organism. So if the organism is stressed, it secretes more pigment. So if it's stressed upon adding the compound, sometimes you see a light coloring of the medium before the fungus stops growing or dies. And that's why this coloration comes from, and that interferes with the color of the viability dye we use. And that's why we use the 20% cutoff and not a concentration, a cutoff value lower than that.
Mm. Yeah, makes sense. Okay.
All right. So in the interest of time, we might just have to move on. We ran a bit over. So is there any more question you want to ask Dimitri before we proceed?
No, I'm all done.
All right, great. Yeah, thanks for that. So very quickly, the next thing is just a biological testing schedule for this year. So I'm guessing this is for any of us who are interested in sending more samples for Wendy's team to test. So I'm guessing, what's kind of like the best and the latest deadline that we can adhere to get everything tested for this year?
Well, for in vitro, we don't have a very, very good deadline, of course, because we can do that the year round. The larvae season will start somewhere in March, April, and end somewhere in September, October. I don't think we are lucky this year to have it all the way until November, because this was really an exceptional time to have that. So if we want to go all the way in vivo, then we need it as early as possible. But if we only want to start with in vitro susceptibility, then we can have it a year around. But preferably somewhere before summer, it should be screened in vitro if we want to have it screened.
You specified the month, because I get confused because I'm living in Australia. It's confusing.
Let's say the 1st of July.
Yeah. So that's sort of like the deadline for if you want in vitro to be done, and there's still enough time to do in vivo, then it's 1st of July this year. Is that correct?
Yeah, that's really the utmost deadline if it needs to be tested in vitro first and then in vivo.
All right, yeah, that's great. Thanks, Mandy.
1st of July.
I think we are good. So there's just any other businesses. I think we've covered most everything. I will organize the agenda for next meeting, so I'll take care of that. I think that's all the items. Sorry, we did run a bit over. Is there anything else that anyone wants to add for this meeting?
Nope, not me.
No, I think for next time it will be good to have the log D values compared to the survival things, etc., so that we can finally make a decision if the log D still is the thing we should aim for, because I think that makes the life for the chemist as well much easier in plans of designing.
I will submit this before, because like with regards to my project, I have to decide like whether we, well, based on what log D trend we see, like what we do next. So I'll probably update everyone on GitHub even before the next meeting.
Yeah.
Yeah.
So just to clarify, do we send? How do we actually send the smiles and all the data to Ben? Is that just, do we send the master list, or do we actually need to create something else on top of that?
Well, if we want to do in vitro and in vivo, and we want to compare both, I think we should send the master list with all the smiles, because we need to know the ones which didn't work in vitro. We need to know the ones which didn't work in vivo, and of course the ones which do work. So maybe it's just best to send the master list with all the data in.
So I'm guessing, so our objective is to for all of us to keep all of our information up to date on the master list, and send that link to Ben to then update us on the log D value.
Yeah, because what Alice is mentioning as well, it's not a very good idea to duplicate all the lists over and over time, because then mistakes might be made and there are differences between the two versions. So maybe it's best just to send a link of the master sheet to Ben with all the smiles in there.
Yeah.
Yeah.
And see if he can calculate.
So there is already a list in that in vitro and vivo tab of compounds that worked in vivo and in vitro, and the smiles are in there. So if Ben can just key in the log D values based on that smile, it'd be easiest. That's of course without the compounds that didn't work. If we want to calculate log D values for compound that didn't work in vitro, then we would have to go through all the compounds really.
The way Ben made it sound, it sounds like it's very quick for him to do it as long as the smileys are there. So I think we just ask him to do it for the whole list that we have, the ones that worked and that didn't, and we can just pull what we need out of it. And then we don't have to ask him every time, oh, we missed this one compound, can you do the log D for us?
So I agree with Kimberly, because it's probably loading the list into a program, and the program calculates everything.
He just copies and pastes it.
Yeah.
So about 1,300 compounds then.
Yeah.
Okay.
We'll ask him if it doesn't work.
Did we screen 1,300 compounds then?
So 3 box of 400, that's 1,200, and then another 100 of more analogs.
Yeah, but he already calculated the log Ds for the pandemic and for the pathogen stasis box, and
I don't think the whole thing. Only the ones that worked in vivo he did.
I think so. Yeah, Wilson. He didn't calculate it the whole. He didn't calculate the whole thing.
Yeah.
Well, then we ask him to do the Fennery moles first, and if he has time left, do the other ones. Because what also was mentioned, that it does matter which class of antifungals you're looking at. So if we do it within a class, that's easier than without the class. And for the other boxes, we need the data on the ASOLs anyways, if we want to cross-ref with the Fennery moles.
Okay.
So in the end, we do need the data.
But for normal series one, definitely is a priority, I would say.
Yeah.
All
Okay.
right.
So who
So
wants
Wendy,
to
please
send
let
the
me know
link
if
to
you
Ben?
need any help with numbering convention with the other boxes that we talked about at the beginning.
I don't think we should give names for the other boxes because it may be easier to look it back in the future when we know, okay, this compound was tested in the pathogen box or the pandemic box, but I don't know.
I think there's a column there in the pandemic, pathogen, and boxes as well, where it was stated from which box, from which, yeah, if it was box A from the pandemic box or box A from the pathogen box. I think it's already in the database, but I will have a look.
Yeah, it is.
Yeah.
So that means that we only have to put them underneath each other and that's it.
Okay. So I will, on my side, on our side, look into double check the master sheet on GitHub again, see what we're missing, hopefully nothing. And then I can, yeah, just tell Ben to go click on the list.
All right. So yeah, that's the number one priority is to just keep the master list up to date for Ben. So that's come from both Wilson from Wendy's team, also from UCIT, and also from Dimitri and Matt as well. So just keep all of our compounds and all of our biological evaluation results up to date, and then we'll send that off to send the link to Ben, who will then use the smart strings to output log the value. So that's the number one action item for this meeting.
Yeah, and for the action list, I will do the MMV compounds, ask the mode of action at MMV.
Yeah,
that sounds great. All right, so I think that's all for this meeting. Thank you, everyone. Sorry, it went a bit over, but yeah, this. I hope.
Thank you for thank you for hosting and for keeping you all up.
Yeah, how dare you?
That's all right. I'm a night owl. This is my normal hour. So yeah, hope everyone who's starting a day have a great day or have a great rest of the day, and everyone else have a good evening, and yeah, I'll see everyone soon.
Have a good sleep.
Bye.
See you.
Bye.